Continuing on with chapter 3, section 4, now we're going to go beyond the plasma membrane. So we've talked about the three parts of the cell: plasma membrane, the cytosol, and the nucleus. We've mentioned the three parts. We've gone into detail about the plasma membrane with the phospholipid bilayer, and then we talked about how in section 3.3 we talked about how items are transported passively and actively across the final barrier actually of the cell. Now we're going, once we get inside into the cytosol and in the cytoplasm, we now have organelles, which basically help with the function of the cell. Right, so now inside the cell, remember the cell is the smallest unit of life. So organelles on their own are not able to sustain life, but organelles inside of a cell, now you have life. Right, so organelles are cellular machinery. Right, they're like the organs of the cell. They have specific functions vital to maintaining homeostasis. There's that word, homeostasis, basically maintaining balance, maintaining your body's functions inside the cell. Some separated from the cytoplasm by the cytosol by membranes, which is compartmentalization. Others are not enclosed in a membrane. The membrane-bound organelles listed here: mitochondria, peroxisomes, endoplasmic reticulum, Golgi apparatus, lysosomes. The reason why they are membrane bound is because their functions could be destructive to the rest of the cell, especially lysosomes. And then organelles not enclosed in a membrane, your ribosomes, those are where we have protein synthesis, and your centrosomes, right? So these are organelles, these are like the organs of the cell, easiest way to think about it. This is a cellular diagram. You see the plasma membrane on the outside. If you cut it open, you would have multiple organelles. And you can have more than one of different type of organelles inside the cell. Ultimately, in the cell, you'll have a nucleus. Notice that wasn't listed as an organelle, as a separate section of the cell. In nuclear envelope and nucleus with a nucleolus in the middle, right, which is going to house your DNA, right, which is, we'll talk about that, but that's not considered one of the nucleus, which is with the nucleolus and nuclear envelope, is not considered part of your organelles. Okay, so everything else inside the plasma membrane is generally, if it's not cytosol, it's considered in the cytoplasm part of the organelles. Okay, so now we're going to go through the different organelles. Mitochondria, known as the power plant of the cell. We're going to talk about a lot about mitochondria when we talk about things such as ATP and the energy of the cell. It is a membrane-bound organelle, provides majority of ATP. Mitochondria has its own DNA, actually, has enzymes, and also has ribosomes. Ribosomes, where we have protein synthesis, enzymes speed up reactions, DNA codes for specific proteins. All right, it has a membrane, as we know, it's membrane bound, double bilayer, smooth outer membrane, inner membrane highly folded into what we call cristae. That's your mitochondria. Each membrane has its own enzymes and proteins. The outer has large channels, allow molecules from cytosol to enter inner membrane, and the inner, more selective membrane transports necessary solutes into the matrix using specific transport proteins. This matrix contains mitochondrial DNA, which code for proteins, and enzymes specific for breakdown of organic fuels by a process called oxidative catabolism. This is how we produce ATP. Oxidative, notice oxidative needs oxygen, oxidative catabolism. Remember, anabolic, build up, catabolic, break down. So breaking down of the nutrients that we've eaten to produce ATP, specifically glucose to produce ATP. All right, so this is the general structure, and we're going to talk about this in more detail when we talk about the whole process of how our body gets energy, specifically when we talk about the digestive system. We're going to get into a lot of detail about how our body not only digests food, carbohydrates, lipids, proteins, but also how it converts that food into energy. So you'll really get into detail when we talk about that system. But that energy production, taking the food we eat, converting it into ATP, that's a whole process involving the stomach, the intestines, the bloodstream. But once you get inside the cell, it involves the mitochondria to produce ATP. Right, so you have this outer membrane, inner membrane here. ATP production is in here, in the inner membrane. Okay, that's your mitochondria. Powerhouse of the cell, ATP production. Peroxisomes, membrane bound, also use oxygen to oxidize organic molecules. Remember what the difference between oxidize and reduce. Electron transfer. If I transfer electrons to something, I'm reducing it. If I remove electrons from something, I'm oxidizing. So it uses oxygen to oxidize organic molecules, produce hydrogen peroxide. Hydrogen peroxide, we use that in everyday world to clean things, but in your body, it's oxidized toxins like alcohol in the liver and kidney, makes them less toxic, and so they can be eliminated from the body. Also helps break down fatty acids. We want to break down those fatty acids. We use peroxisomes. So peroxide, hydrogen peroxide, peroxisomes, right? We do not want hydrogen peroxide just floating freely in our cells. That's why we have these peroxisomes in membrane-bound organelles. They also can synthesize certain phospholipids. Remember those? Phospholipids, which help make up the structure of these membranes, specifically the plasma membrane. Ribosomes, tiny, granular, non-membrane bound organelles. So non-membrane bound. And this is, as I've mentioned before, the site of protein synthesis. And we're going to talk about this in a lot of detail when we talk about protein synthesis in DNA, RNA, and DNA. And this is where a ribosome is composed of large and small subunits, each made of ribosomal proteins and ribosomal RNA. They float freely in the cytosol. And we're going to find that when they bind with a molecule of RNA, we have this process called translation that happens. And in that process of translation, we can convert RNA into its we can translate RNA into a protein, into amino acid chain, which is then into a protein. We kind of talked about that in the last chapter. This is the location of protein synthesis. Now we're going to go into the endomembrane system. You see that there is multiple slides about this, but this can get a little complicated. Remember, we have vesicles. We talked about vesicles and we talked about endocytosis, bringing liquids and solids into the cell. Also, in regards to exocytosis, bringing liquids and solids out of the cell. Right, so our endomembrane system forms vesicles that exchange proteins and other molecules, synthesize, modify, package molecules produced within a cell. Components are of the system are a plasma membrane, nuclear envelope, endoplasmic reticulum, Golgi apparatus, and lysosomes. These all work together. Endoplasmic reticulum, large folded phospholipid bilayer, similar to the plasma membrane, continuous with the nuclear envelope. You have rough endoplasmic reticulum. The reason we call it rough is because there's ribosomes attached to the surface, and you have smooth endoplasmic reticulum, which has no ribosomes attached to the surface, right? And this sort of provides a pathway for vesicles and things to pass through. The rough endoplasmic reticulum, specifically, so products will enter the rough endoplasmic reticulum lumen. Incorrectly folded polypeptide chains detected, sent to cytosol for recycling. Most proteins enter the RER, rough endoplasmic reticulum, are for transport outside of the cell. Packages, what happens in the rough endoplasmic reticulum is things get packaged into transport vesicles made of phospholipid bilayer, same phospholipid bilayer, such as a cell. This is making it into a vesicle, which we talked about with endo and exocytosis, and is sent to the Golgi apparatus for further processing. Also, the rough endoplasmic reticulum produces membrane components for membrane-bound organelles and plasma membrane, including integral and peripheral proteins. Your smooth endoplasmic reticulum has no role in protein synthesis. Remember, the rough endoplasmic reticulum has the ribosomes connected to its surface, so it's involved in protein synthesis. Smooth, not so much. No ribosomes connected to the surface. It's more so for storing calcium ions, which are pumped out of the cytosol for future use. Detoxification reactions and lipid synthesis. Right, so manufacture majority of plasma membrane phospholipids and cholesterol. Picture of your endomembrane system, your rough endoplasmic and smooth endoplasmic reticulum. Notice this around the nucleus. All right, and these provide pathways, help make vesicles, package proteins, phospholipids that are to be transported different parts of the cell or out of the cell. We also have the Golgi apparatus between the rough endoplasmic reticulum and plasma membrane. These flattened membranous sacs filled with enzymes and other molecules. Proteins and lipids made by the endoplasmic reticulum are further modified, sorted, and packaged for export in the Golgi. So it takes things that are made, packaged by the endoplasmic reticulum, and goes to the Golgi apparatus, and we just they can be modified more. Products packaged in the Golgi can be then secreted from the cell by exocytosis, joined to the plasma membrane, or sent to the lysosomes for a destruction, actually. So we have a vesicle going from the rough endoplasmic reticulum here to the Golgi apparatus for further processing, packaging, new vesicle forms, and leaves from there. You've probably heard of the disease known as cystic fibrosis. Very destructive disease. My wife works at the Children's Hospital of Pennsylvania, and therefore comes across, she works on the floor that deals with this disease. Some cells are missing the missing protein component of chloride ion channel, causes deficient chloride ion transport in lungs. You need chloride ion transport, affects your digestive, your integumentary, your skin. It results in abnormally thick mucus, blocks airways, causes digestive enzyme deficiency. Mutation causes chloride channel protein to misfold slightly in your rough endoplasmic reticulum. So it's a mutation that affects your rough endoplasmic reticulum. The chloride channel protein that's created, or that's folded, processed by your rough endoplasmic reticulum, doesn't fold properly. Therefore, protein destroyed, even though it would be functional if inserted into membrane. Disease is caused by overprotective rough endoplasmic reticulum. Remember, we talked briefly, the rough endoplasmic reticulum, if something is a protein is not folded correctly, it destroys it. Well, this protein is needed so that chloride can get into and out of your cells. So it's a very destructive disease. Lysosomes. Organelles responsible for digestion of worn out cells or cellular components. They contain digestive enzymes, also have membranes. Therefore, we don't want the insides of the lysosome just floating inside the cell. They contain acid hydrolases. So when part of the cell is worn out, it needs to be broken down, destroyed, it goes to the lysosome. Macromolecules broken down into smaller pieces and released to the cytosol for disposal or reused to manufacture new macromolecules. Okay, so that's where we can have things broken down. And this is just a summary of your endomembrane system, so you can review that on your own. The cytoskeleton, as we continue on inside the cytosol, the cytoskeleton, dynamic structure changes function based on needs of the cell, just like the skeleton in your body. The cytoskeleton sort of gives the cell its shape along with the plasma membrane. Okay, creates sort of like an internal framework, provides strength, structural integrity, anchoring sites, allows for cellular movement, motor function, helps with phagocytosis, and contraction of muscle cells. Right, there are 3 types of filaments in your cells. You have actin filaments, intermediate filaments, and microtubules. This is like the structure, the skeleton of your cell. Actin filaments are the thinnest filaments, composed of 2 intertwining strands of actin subunits. You may have heard of actin and myosin in referring to muscle cells. So actin is a type of microfilament. Actin and myosin in muscle cells when they contract, allow for your muscles to contract and expand. So you may have probably heard of those, which we'll talk about when we get to the chapter on the muscle system. But the actin filaments provide structural support, bear tension, maintain cell shape, involved in cellular motion when combined with the motor protein myosin. Right, that's the thinnest filament. We also have intermediate filaments made of different fibrous proteins, such as keratin. Keratin, it's in your skin, it's in your nails. Keratin, in your hair. You've probably heard of keratin. Form much of the framework of the cell. Anchor organelles in place. We don't want organelles just necessarily floating around the cell. We want them kind of in a specific location. help organelles and nucleus maintain its shape, help cells and tissues withstand mechanical stress. So that's your intermediate filaments. And then we have microtubules. That's the largest filament. Hollow rods composed of what we call tubulin. Can be rapidly added or removed, allow for size and shape changes within the cell, help maintain the cell, keep organelles in alignment as well, similar to the intermediate filaments. Have the motor proteins dynein and kinesin, which allow transport of vesicles, right? Transport vesicles is necessary for your endo and exocytosis. Microtubules. And this is images. These are images and listing of the different functions of the 3 different types of cytoskeletal filaments. Microtubules extend out from the centrosome, which is a gel matrix containing tubulin subunits. When cell is not dividing, we're going to talk about cell division. Cell division is going to be very important when we talk about things like meiosis and mitosis. It's going to be very important. Cells have to divide. That's how we grow. Remember, we all started as a single cell, and now we are all made up of millions of cells. So cells divide constantly have to divide also for replacing cells that have worn out or dying or dead or repairing. So cell division is very important. When a cell is not dividing, the centrosome is microtubule organization center located close to the nucleus. Microtubules, those are your biggest filaments. Pair of centrioles is critical for cellular division. You also have basal bodies, modify microtubules on the internal surface of plasma membrane where flagella and cilia originate. Flagella and cilia, which are important for movement, come from basal bodies. So centrioles and basal bodies are centrosomes. These are just some important locations for where the microtubules extend out of. Okay, I guess I numbered these slides a little bit off. Cellular extensions are formed by inner framework of cytoskeleton. We have microvilli. Cellular extensions, extending beyond the cell. Well, extending out, sort of out of the plasma membrane. Microvilli, cilia, and flagella. You probably have heard of at least 2 of these, probably. Cilia are very important. My daughter actually has an illness, a genetic illness, in regards to her cilia not functioning properly in her airways, which we'll talk about. But first, we have microvilli, depending on how you want to pronounce it. Finger-like extensions of plasma membrane with finger-like extensions of plasma membrane with actin filament core. Okay, actin filament, the smallest filaments. Increased surface area of cells and organs with specialized for absorption. We'll find these in the intestines, especially. We want to sort of increase the surface area so that in the intestines, so that food, the nutrients from the food can be absorbed quickly into the bloodstream. Microvilli. Cilia. We have hair-like projections composed of microtubules and motor proteins. So bigger tubules, bigger filaments, microtubules and motor proteins. They move in unison to propel substances past cells, found in great numbers on each cell. All right, cilia. You'll see this especially when we talk about when you get to the chapter on the pulmonary system, you'll see cilia are very important for moving mucus and any type of debris along in your airwaves. And then we have flagella, which is solitary. That means one, longer than cilia, found only on the sperm cell. Right. So in the male, on the sperm cell, beats in a whip-like fashion, propels the entire cell. This is what helps sperm to swim. Flagella and cilia are very similar, structurally similar to centrioles, except they contain two central microtubules not found in centrioles. Right. So basically, flagella is basically this microtubule, very similar to cilia, just really long and allows the cell to be propelled. Right. This is showing you the structure of cilia and flagella, made up of microtubules. And this is just showing the location of cilia and flagella. Respiratory tract, female reproductive tract, where we find cilia. Sperm cells, where we find flagella. Microtubules. And this is the disease that my daughter has, I mentioned. Rare genetic disorder, defect in one or more protein components of cilia and flagella. Affects the respiratory passages, middle ear, uterine tubes. It can lead to a buildup of mucus in the lungs. She has an increased risk of infection, cilia dyskinesia. Kinesia. Right? So basically what happens is her cilia, she doesn't have any flagella, she's a female, but her cilia basically doesn't function properly, and therefore it's very difficult for her to move mucus. And we noticed it in her airways. And so she actually had mucus buildup, and she was delayed in her speaking because she couldn't hear that well. And so we had to get like ear tubes put in. We had to get mucus like sucked out of her ears, out of her airways. It's definitely gotten better now that she's gotten a little bit older, but it's something that was very, very difficult to deal with early on. So her nose would just run. Her, mucus would come out of her ears from time to time. It was pretty difficult. And then they finally diagnosed it. But yeah. All right.